So it looks like the people in the waiting room are in our official Zoom room. So this is Brian Cunningham saying welcome to the IGB seminar for the month of November. And it's my pleasure to welcome my friend and colleague, Professor Benjamin Miller from the University of Rochester Medical Center. Back when I first invited Professor Miller, I thought for sure you'd be able to come in person by November, but it looks like, of course, we're still in COVID pandemic, and thanks very much to Professor Miller for still agreeing to come and give a seminar virtually. Professor Miller earned his PhD from Stanford University in organic chemistry. He trained as a postdoc, as an NIH postdoc fellow in the Department of Chemistry at Harvard University under Stuart Schreiber's group. Currently, he's the Dean's Professor of Dermatology, Professor of Biochemistry and Biophysics at University of Rochester Medical Center. And I know that he'll also talk about some of his work, not only as a professor and researcher, but also as an entrepreneur. So welcome, Ben Miller, and looking forward to your talk today. The floor is yours.
Thank you, Brian. It really is a delight for me to be visiting Illinois virtually. I would obviously much prefer to be there in person, but I'll take what I can get, and hopefully we'll have an opportunity to come and see you all in real life at some point when we're past all of this. So today I'll be talking about how we're applying photonic sensors and other kinds of optical sensors to decoding complex biology. And I guess I should preface my talk by saying that because this is a fairly mixed audience, I want to talk about some biology and some sensors, and I know that is going to risk annoying people on both sides of the aisle, biologists and engineers, but hopefully there will be enough aspects of both that it will be interesting. So a lot of biological research today is focused on big data, and really how do we manipulate large amounts of information about biological systems, whether that's genomic data, proteomic data, metabolomic data, whatever omics you want, and use that to understand how organisms grow, develop, and interact with the outside world. But one of the problems is our world is an analog universe, right? We're made up of things that are not digital, although I guess in some fields of research, people like to look at performance as digital. But really, we need some way to interface between the analog world and all of that big data that we want to manipulate to understand biology. And so when we're feeling in a pretty grandiose mood in my group, what we say is that what we work on is the interface between the universe and data. Building sensors that can allow us to take that information and transform it into the digital domain so that it can be manipulated down the line. And so we do that using optical techniques. And the basics of how biosensors work is we have some sort of optically responsive material. Typically that's coated in some fashion with a capture molecule, although in some cases it needn't be. But what happens then is the analyte of interest, a protein, a piece of DNA, a piece of RNA, will bind to that capture molecule. And what happens then is we get a signal characterized by either an intensity change, a spectral signature, or a shift in a spectral feature. And as I said, we make optical sensors. You can think about similar ideas in the field of electrical sensors, but I'm going to focus on these today, particularly on spectral signatures and shifts in spectral features that are characteristic of detection of a molecule. Now, what's really interesting about this and what differentiates optical and photonic sensors from methods that have gone before, like ELISA assays or bead-based assays like Luminex, is that because we can address different areas of an optical sensor individually, either using individual inputs and outputs or in an imaging mode. What that allows us to do is, for the first time, we can detect as many different things as we want simply by having different unique detection areas. And that's something that really you can't do with an ELISA or even with something like a Luminex. In both of those techniques, you reach an upper limit on what you can do in terms of, in the case of ELISA, your ability to coat antigens and process them in a 96-volt plate. In the case of a Luminex system, there's an upper limit on what you can do without worrying about cross-reactivity of all of the secondary antibodies that are necessary to make the tool work. So over the last couple of decades, a longer period of time than I necessarily want to admit, my group has worked alone and with collaborators to develop a series of different platform technologies to detect biomolecules using changes in the optical properties of materials. Obviously, in a relatively short seminar, I won't have time to go through all of these, but a couple that I'm going to focus on today are, in particular, probably what's the least attractive of the pictures that I have on this. This is a sensor that's based on reflectivity on a silicon chip. And then another one that's actually not on this timeline, because we didn't really get started on this until around 2016 or so, based on integrated photonics. So let's start with this reflective sensor. So if you've ever taken a silicon wafer that's highly polished, you note that it's reflective. And what we do is we make use of those reflective properties, and actually more precisely, the anti-reflective properties, to be able to directly detect biomolecules in a label-free multiplex manner. And so to begin to introduce this technology, let me make a little bit of an analogy. If you are walking through a cityscape in the daytime and you look up at the buildings, because of all of the ambient light, it's impossible for you to know what's going on in those buildings, right? All of the light that's external to the building is reflecting off the surface, and you don't know who's in what office or what's going on. But if you come back at night, when all of that ambient light is gone, you now have a much higher contrast between what's in the offices and the external environment. And so I can see which offices have lights on in them. I might be able to pick out some furniture, some people walking around. My ability to detect those very small, subtle events is significantly improved by decreasing the ambient radiation. So the same thing is true if I think about making a sensor based on reflection. If I focus on a starting condition where I'm close to zero, or as close to the 0 point in reflectivity as I can get, what we know from basic junior high school math is that if this number is close to zero, then any change relative to a zero is going to be a very large number and easily detectable. So all we have to do is create a condition on a chip where our background reflectivity, that is the reflectance and the absence of what we're trying to detect, is close to zero. And it turns out that the principle of how to do this has actually been developed in classical optics going back to the late 1800s. It's just nobody really found a use for it. And this is based on equations worked out by Fresnel and ideas of interference, in that if we have a coating on the surface of a silicon chip, and we think about light reflecting off the front surface and the back surface of that coating, we can set up a condition where these rays completely constructively or destructively, interfere with each other if we carefully control the input wavelength, the angle, and the thickness of the material. So, all right, input wavelength we can control based on selecting something like a helium neon laser line, which is controlled by quantum mechanics. Perfect every time. The input angle we can control just physically, just by manipulating the exact position of that input laser light. And then if we're thinking about a coating like SiO2 on a silicon surface, and if we also consider that the refractive indices of your typical biomolecules are similar to the refractive index of silica, we can set up a thickness condition where this absolutely gives you that 0 point, where the input angle is around 70.6 degrees. The wavelength, again, helium neon laser line, and the thickness is around 1400 nanometers. So that gives us a technique that we call array imaging reflectometry. And this is the idea that we start out with biomolecules immobilized in a microarray format on a silica surface on a silicon chip. No light comes out in the absence of target. In the presence of the target, we begin to get partial reflection as we degrade that antireflective coating. So this is different from other techniques like surface plasmon resonance or reflective interference spectroscopy, in that we're really trying to create a zero and then perturbing it rather than looking at a minimum and watching for a shift in the minimum. So, okay, that's the idea. How well does this work? Well, initially to actually validate the tool, the technique, what we did was we photolithographically patterned posts in a silica surface on silicon and precision etched those so we could control the height of the post above the background. And what you can see is that you can easily visualize a 700 picometer step. You can see a 75 picometer step still quite easily. And even down to a 10 picometer step for these posts is easy to pick out relative to the background. Now, thinking about that, what that means relative to biomolecules we might want to detect. Human serum albumin is about a 7 nanometer diameter spherical molecule. Glycine, 4/10 of a nanometer. Water is 2/10 of a nanometer. That 10 picometer step is 1 pixel down here at the bottom of the screen. Okay, so obviously we're not actually imaging sub-wavelength changes in the thickness of the surface. What we have is we have a CCD camera. We've got our laser and our chip. The CCD camera is made up of pixels, and each pixel in that CCD camera is averaging over a particular area on that surface. So what we see is an average where you're plucking an atom out here and there over the space imaged by a CCD pixel, and that corresponds to that average 10 picometer change. Okay, but still, what that means is that we can detect sub-monolayer of water changes in thickness. In principle, if we can control all of these variables. So here's how this actually works in practice if we're doing an experiment. What I'm showing here is a graph of experimentally derived thickness references where we've calibrated all of these by spectroscopic ellipsometry. You can see that we have a parabolic fit to those data. And if we make a microarray, what we do is we start with everything as close to zero as possible. That includes our probe molecules, the antibodies or antigens that are going to be detecting targets, as well as a control that we have spaced around all of the probe molecules. For quite a long time, we've used antifluorescein as a control molecule. I'll mention in passing that we're beginning to get away from that and looking at other controls. I can follow up on that for those who are interested. But for many of our experiments, this antifluorescein works quite well because you shouldn't have any fluorescein in your bloodstream, for example. Then if something binds, what you're going to see is that for those molecules, you have an increase in thickness that corresponds to an increase in intensity. And by comparing that to the change, if any, in the intensity of the control spot, we can directly derive how many angstroms of material was deposited very precisely. So what I'm going to tell you about today is how we applied this technology and others to detecting both antibodies to upper respiratory viruses, as well as antigens and detecting the viruses themselves. Obviously, this is a timely thing, and you'll see how some of the flu work that we were doing primed us for working on SARS-CoV-2. And actually, I'll focus the bulk of my talk on our SARS-CoV-2 work. But this really grew out of wanting to be able to use antigen arrays. In the case of flu, that's the flu hemagglutinin, for detecting antibodies, profiling the immune response in humans and animals of various kinds, either for basic biology or to understand vaccine efficacy. And then antibody arrays to do direct detection of viruses as a diagnostic method, but also as a way to understand the immunological relationships among viruses. So let's actually start with the virus detection piece for flu. And this is actually work that Hanyan Zhang, who many of you know, did as a graduate student in my lab. This is what we call a crowd on a chip approach to detecting flu viruses. And what we did here was we teamed up with Patrick Wilson's lab at the University of Chicago. Patrick's group takes subjects who come in and get vaccinated and then has them come back after a couple of weeks. They take a blood sample, and then after isolating plasmablasts from those blood samples, they clone antibodies from individual subjects. So they have a huge library of these things. And just like any of you who are participating in this call, if you get vaccinated against the flu, you'll each have a slightly different response to that vaccine than I will, or to what your colleagues will have. What that means is that each of these human monoclonal antibodies will have a slightly different pattern of affinity to different flu viruses. And so if we, for example, spot 90 of these on an array, that's a combination of 90 monoclonal antibodies where each antibody is spotted twice, so we have duplicates, and then surrounded by those anti-fluorescein proteins, so we always have our reference. What you can see is that we can readily distinguish a flu strain that's an H7N3 flu, and if you're not familiar with this terminology, that's the H is the hemagglutinin, N is neuraminidase, and then the numbers just say which homologous form of hemagglutinin and neuraminidase it is. Easily distinguish that from an H1N1 virus, even just by eye. But as it happens, we can even distinguish very closely related viruses. These are two H7N9 viruses, A. Anhui, H7N9, and A. Shanghai, H7N9. These differ only by 6 amino acids in their hemagglutinin, and by plotting the response of the antibodies on these arrays, we can easily tease apart even these very closely related strains. So that was a preliminary validation of this approach. The next thing that we wanted to do with this was to say, all right, let's say that we look at the vaccine strains of influenza over an extended period. Can we actually use data from one of these arrays to develop antigenic relationships among these different strains, and use that to understand how efficacious vaccines were in different years, but then also use that to then make predictions about where the flu was likely to go, as well as use it in ways to relate unknown flu viruses to what's already known. So here's an example of how Hanyan did that. This is with a 115-plex, again, human monoclonal antibody array. What Hanyan did was screened a whole series of different H3 flu viruses. These are all vaccine strains, comparing them to the control. And here, in order to be able to visualize this data, what he did was a data reduction method that put everything into a two-dimensional plot called an antigenic map. And this is a standard approach that's used in immunology for characterizing the relationships among viruses. And what you see is that on this plot, the monoclonal antibody array using the AIR technology is in red. We've compared that to the traditional technique, ferret hemagglutination assay, in blue. and then whole genome sequencing in black. And what this allows you to do is see how closely related different viruses are. So where this is for the sequencing method, this bar corresponds to a 5 amino acid difference, or in our case, what we call just a 1 antigenic distance, which is an arbitrary unit, but corresponds reasonably well to a hemagglutination antigenic distance. And you can see that some of these cluster very well. It's Brisbane 07, Uruguay 07. We didn't have ferret data for this one. Wisconsin 05, that clusters very well. But there's some really interesting differences. For example, we put Switzerland 13 way out here, so does sequencing. But the ferret assay, again, the traditional method for understanding flu, is putting this much closer to Texas 12 here. Now, the reason that that's important is if we look at the efficacy of different flu vaccines using and comparing it to this antigenic cartography, in 2008, 2010, the vaccine virus was primarily this BR07 strain. The circulating virus, that is the ones that people were actually getting infected with, was either this Brisbane 07 or Uruguay 07. Okay, and those are pretty well clustered, so it makes sense that this would have a reasonable efficacy. But if we look at the 2014-2015 season, here the vaccine virus was Texas 12, okay, down here. The circulating virus was Switzerland 13. And if you're relying on ferret assays, you'd say, that's not too bad. But if you look at either genomic sequencing or our assay, it's way far away antigenically from that Texas 12. And indeed, if you look at the vaccine efficacy for that year, it was only about 19%. So obviously this is correlation. This is one initial, interesting observation. It's not necessarily going to be predictive and reliable ad infinitum, but I think it's an interesting observation that we have been developing our understanding of flu and our predictions for what's going to be useful a vaccine largely based on these ferret assays. And while ferrets are incredibly useful as research animals, everyone knows that their immune system is not the same as a human immune system. Whereas with these human monoclonal antibody arrays, we can have a fully humanized understanding of what's going on with the virus. So potentially useful. More recently, we took that information and applied it to a mock pandemic exercise. This is, again, Hanyan's work with many other people. One of the things that I like about this paper is all I need is for everybody who's an author to cite it once, and my H-index will go up. But this was a massive effort nationwide led by the network of flu research centers, basically given the charge by NIH that let's pretend that 2 different strains of dog flu have suddenly become pandemic. Figure out how the network is going to respond. And so the first thing that Hanyan did was took his array and said, okay, which virus do these most resemble? And it turns out that the closest match is what's known as the mother clone for influenza vaccines, Hong Kong 68 H3 virus. And from that, and I should say we also compared to this Wisconsin 05, it's not as close as a match as Hong Kong 68. And from that, we can then pull out cross-reactive antibodies that are potentially useful as therapeutic agents, but also as ways to begin looking for epitopes that are useful for vaccine development. And then finally, in the flu regime, I want to mention that 2 things, actually. Hanyan, in the last part of his work, also looked at understanding differences between bird and human infecting viruses using glycan arrays. So that's another option that you can use for printing these. Here, the alpha-2,3 linkage glycan and the alpha-2,6 linkage glycan determine whether something is a human infecting virus, that's alpha-2,6, or avian infecting virus, alpha-2,3. And we were able to verify discrimination of an H1N1 pandemic virus, that's a human infecting virus, selectively bound the alpha-2,6 glycan, and H13N8, that's a strictly bird virus, bound explicitly the alpha-2,3 glycan. So another approach for looking at viruses. Of course, as I mentioned at the outset, antigens are also of interest. And so this was an 18-plex hemagglutinin array that Joe Buchkowski and my lab developed for looking at understanding serology. So looking at antibody response, both in humans and in this case, in birds. I won't dwell on this just given the time, but basically what this showed is that we could do multiplex serology on a bird sample, which is something that is very challenging to do otherwise just because of sample volume requirements. So this flu work had us pretty well primed for when we all started seeing charts like this at the outset of March. Well, mid-March is when we really started paying attention to it, I guess, and started worrying about the COVID-19 pandemic. And even more importantly, started really thinking, how do we make the Zoom meetings stop? So we thought about how we could use these array technologies to understand flu serology and test for antibodies to SARS-CoV-2. Now, obviously, serologic testing has proven to be a really important part of a response to the pandemic. This is understanding what antibodies are developed in a patient following active infection. Many people have had this idea, obviously. There are, as of when I made this slide in May, there were about 170 antibody tests reported or marketed. But one of the problems is that many of them were not particularly high quality. So we started with the goal of being able to quantify, so do better than a binary response, discriminate between SARS-CoV-2 and common cold viruses, and then also from the outset, we wanted to work with partner companies so that we could distribute this and get it dispersed globally once we had something that worked. So we started our COVID-19 effort on March 13th. I'm going to tell you fairly quickly about two different approaches that we've been taking. One of these is based on the air technology. The other is based on a disposable photonic sensor. The air technology, we developed that and then have been collaborating with Adarza Biosystems, which is a company that spun out of the lab to commercialize this. The disposable photonics, we're collaborating with Ortho Clinical Diagnostics to get that put into people's hands. So looking at relationships among coronaviruses and thinking about what we might want to have on our array, obviously SARS-CoV-1, the original SARS, and SARS-CoV-2 were important to have. But I think even more important were the different circulating coronaviruses. HKU1, OC43 in particular, which are common beta coronaviruses that are in the same general lineage as SARS-CoV-2, but then also alpha coronaviruses like human coronavirus 229E. If you think about the antigens, I know many of you have worked on SARS-CoV-2, so I don't need to explain this in a lot of detail, but just to review, the primary antigen that we were interested in at the outset was this spike protein. It's what gives the coronavirus its shape and its name, and is the protein that makes direct contact with the ACE2 receptor on human cells. The spike protein can be further divided into different subdomains. So our antigens include the full-length S1 plus S2 ECD, the S2 subunit, the S1 subunit, and then the RBD or receptor binding domain that is the direct contactor of ACE2. Along the way, it became clear that many groups were also finding the nucleocapsid protein, or N, which is this guy on the inside, to be useful for looking at antibody response to SARS-CoV-2. So in some of the later data I'll show you, we've also included N. Looking at the sequence homology for these different respiratory viruses, this is now just looking at the receptor binding domain for the different coronaviruses and then comparing that to hemagglutinins, which is, yes, a crazy comparison, but there you have it. For flu, obviously, there's no real homology between hemagglutinin and RBD. And looking at the different RBDs for the coronaviruses themselves, you can see that SARS-CoV-1 and 2 are very closely related. Really not much homology to MERS coronavirus, and certainly much less to the circulating coronaviruses. But nevertheless, interesting to consider whether there's any useful cross-reactive response that we might be able to see. Here's the initial array that we printed with these different coronavirus antigens. I apologize, this table is a little difficult to read. We got all of the coronavirus antigens from cynobiological. These are produced in baculovirus infected insect cells. The hemagglutinins, the Brisbane HA and the Wisconsin H3HA, those also came from cynobiological. The California H1 hemagglutinin was donated to us by Florian Kramer's lab at Mount Sinai. So in these arrays, each antigen is printed in 6 spots, surrounded by our anti-fluorescein printed at 3 different concentrations. As you can see, we've got, this is full-length spike for HKU coronavirus, S1 of MERS, RBD of MERS, S1 of SARS-1, RBD of SARS-1, S1 plus S2 of SARS-CoV-2, there's S2, S1, and RBD of SARS-CoV-2. Human coronavirus, full-length spike, 229E and OC43, and then the 3 different flu antigens. We have a blank, and then we repeat each array. So one thing to note on this technique is that if you think about each pixel, each spot has around 120 pixels. There are 6 spots for each antigen, and then each array of 6 antigen spots is printed twice on the broader array. There's a lot of data that comes out of each one of these chips. The chip itself is 5 by 6 millimeters, roughly. In the lab scale device that we work on, we just attach these to things that look like gel combs, and then those can be manipulated a strip at a time in 96 well plates. So actually pretty easy. So you basically just put your sample in the plate, dip the chip in there, wait however long you want to wait. For these arrays, antigen arrays, it's we typically let it go an hour. We really don't need to let it go that long. But then you pull this out, rinse it in image, and you're done. So after printing the array and doing some initial optimization of the print conditions, we looked at first a background matrix. This is fetal bovine serum and pooled normal human serum pre-COVID. So what you're seeing here is some of this is a little bit issues with print conditions on these early arrays. This is one of the circulating coronaviruses, and it printed a little bit thick. But others are just response by the pooled normal human serum to flu hemagglutinins, other circulating coronavirus antigens, that sort of thing. But then if we expose this to an anti-RBD polyclonal at 1 microgram per mil, you can see that the SARS-CoV-2 antigens light up. Actually, they saturate the camera for all of the RBD containing antigens. That's S1 plus S2, S1, RBD, not the S2 domain. So that behaves as you would expect. If we look at this quantitatively, I'll show this to you actually in a couple of different ways. Here's the full array, again, for this polyclonal. As you'd expect, there's really not much response to the flu antigens, and all of these values are in angstroms, titrating from 1 microgram per mil down to 64 picograms per mil. You can see that here again, we had some formulation issues to address in that the 229E spike, even in the matrix, is giving a pretty thick response. So we subsequently thinned this one out a little bit. That's pretty straightforward to do, actually. And likewise, S1 plus S2 ECD started a little thick. But otherwise, you can see that there's strong response, as you would expect. A little bit of cross-reactive response to 229E. It's just a sticky protein, and then this titrates out also with well-behaved responses. If we look at that in a little more usual format, these are now fit to a standard 4PL fit or Langmuir type curve. What you can see is that all of these are analytically well behaved. The limited detection for this assay, this is not optimized at all, is around 10 nanograms per mil. Although certainly there is value in some instances in ultra-sensitive array assays for coronavirus antibodies. For the application that we envisioned, we really didn't need better sensitivity than this, so we did not optimize beyond this detection limit. So with that initial data showing that we could actually make the array work with a polyclonal antibody, the next question was, well, what about human samples? So we initially had, I guess, from a public health standpoint, the happy case; from the researcher standpoint, the unhappy case that we had no patients really that we could access that had COVID-19 to get serum from. So we turned to lab volunteers, took serum samples from everybody in the lab, including me. One of these is mine, and started looking at them on the array. And what you can see is that for most of these, I should say here, I'm just showing the SARS-1, SARS-2, and MERS antigens in this graph. Most of these are pretty low, not much of a response. There are some where you see some interesting things with a couple of antigens. For example, these two have saturating responses on the SARS-CoV-2 RBD. Both of these subjects initially said they had no known incidence of COVID-19 at all. But subsequently, we found that this person was this person's spouse, and this person had actually been in contact with somebody who'd had COVID-19. So, okay, these people probably actually had COVID-19. Same thing with these two samples, this one and this one. Again, no history of COVID-19, but you can see a similar sort of antigen response pattern. And it turned out that both of these people, again, spouses, had traveled to Seattle in late February. So potentially they both had at least mild disease. But the real issue was, without a known sample, where's the threshold? So we really had to wait a bit to be able to assess whether these were true positives or not. And about second week of April, we got access to a series of convalescent COVID-19 serum samples. Initially, just 15 acquired via the U of R Medical Center Healthy Donor Protocol. We've subsequently amplified that, but I'll just stick with talking about these 15 samples. These were all at least 27 days since their symptom onset, so past active disease, broad age range, good balance of males and females. One interesting thing with this is that 5 of the subjects had been classified as COVID positive, but actually had not had a positive PCR test. And you'll see that actually shows up in our arrays. Here again, same array. And what you see now, this is further optimized printing conditions a little bit since what you saw a couple slides ago. Negative donor sample looks like this. So response to some of the flu antigens. Convalescent serum, now we're lighting up those SARS-CoV-2 antigens. Looking at this quantitatively, it's easy to pick out the subjects who had null or no positive PCR tests because they show no response. They really did not have disease. So we were able to reclassify these subjects. But you get a broad range of signals. Here, we're starting to see a lot of antigen response on really all of the SARS-CoV-2 antigens. For the ones with the highest titer, we also see some cross-reactive response to the SARS RBD. as well as here we still had that issue with a little bit overprinted 229E spike. But you can now pick up relationships among these subjects' response and their pre-existing responses to flu antigens as well as the other coronaviruses. We looked at those at a couple different dilutions. Actually, the further diluted material gives us analytically cleaner data. And so we, subsequent to this initial set of experiments, have been doing everything with the 1:20 dilution. If you want to look at this in terms of diagnostic capability, again, we had some of these misclassified. So actually our sensitivity is not great. That's not a surprise because this still includes some of those false, what were really false positive samples that we counted as positives. But our specificity is outstanding. And with more samples, we've been able to improve this. But statistically, very clear differentiation between the positives and negatives. Another important confirmation of the analytical performance of the assay is how well it compares to a reference standard. For this, we used a singleplex ELISA assay, where collaborators looked at IgG titers in microgram per mil for 4 different antigens. One of the things that's amazing here to me is just what the titer is for some of these responses. So OC43, here's someone who has, actually this person has 300 micrograms per mil of anti-OC43 specific antibody in their serum. So that person has had a pretty nasty cold at some point. Pretty interesting to think about. But taking these IgG specific values and plotting those relative to our air results, keeping in mind that the air results are antigen specific, but total antibody, while the ELISA is antigen specific and IgG specific, we still get really nice correlation between the two techniques, reasonable R-squared values, particularly given that they're not measuring exactly the same thing. So that was encouraging. We can also get at specific antibody titer by adding another step where we throw in either an IgG or IgM. I won't dwell on this, but we can pull out those values quite easily, again, in an array format, getting at IgG or IgM specific responses for all of the different antigens. So after we developed this, we, as mentioned, wanted to hand this off to a company that commercialized that. So Adarza Biosystems took our antigen formulations and developed an array expanding what we had now on this Ziva platform, which is their name for their automated version of the air assay. This uses a consumable that looks like this, that's fully enclosed, has the chip within it. So that's 2.5 microliters of serum diluted 1:20 is enough for working in this chamber. You can operate the whole thing in a 96-well plate. And so what one does is you add samples, you incubate however long you want. For other assays, for example, the company is now selling a 40-plex cytokine array. You can also add a reagent to enhance the sensitivity to get down to single picogram per mil detection limits. For this assay, we just incubate it and then throw it in the machine. The machine does everything from rinsing the chips to measuring the arrays to reporting back quantitative information. So this is just the arrays again. It's an expanded version of what we had. And what we see is that for a series of samples, you get very good information, being able to discriminate positive and negative samples, repeating our samples that we had run by hand. Now with the Ziva system, we get very similar results. And importantly, we can now very easily in this 96-well format do titrations to look at measuring actual titers of all of these responses. And we can start to look at correlations to see whether there are interesting, does it matter whether someone had a pre-existing high antibody titer to, for example, HCOV 229E, does that correlate to the response in SARS-CoV-2? And by the way, this was another sanity check for us. We had RBD from 2 different manufacturers. This is a correlation between a cytobiological antigen and another one made by Linco, and you can see that there's an R-squared of 0.99 if you round up. So very good correlation there. We've looked at monoclonal antibodies. So another potential use for this tool is as a way to examine monoclonal antibodies that might use as a therapeutic agent. These are some from active motif. Many of these respond very well to RBD. You can see that one of them that they sent us actually was not good. And it turns out that was something they were able to subsequently confirm. But it's a nice way to very quickly assess the relative affinity and specificity of these monoclonals for different antigens. And then one of the really interesting things that we're finding now is that some of the people that we acquired serum from early on in the process had not had COVID-19. Obviously, as time passes, more people are getting this. And so we're now able to get some of these people to come back and give us more serum samples and we can look at before and after, as well as now following convalescent COVID-19 patients over time using this methodology. So I'll just show you a couple of these samples that are interesting. Here are three, two females. One became ill end of June and then gave us samples July 1st and October 22nd. Another one who became ill in early October gave us a sample 12 days later, had very mild disease. And then this person's spouse who had no symptoms and a PCR negative response for COVID-19. And this is a super busy slide. I won't dwell on it, except to show that one of the things that's interesting through this period, these are all the flu antigens. And you can see that, for example, this person, when you look at their response to the H3s in particular, you can see a significant increase in the antibody titer. And going back, yes, this person had a flu shot between when those two samples were donated. But then looking at these subjects that we've been following over time, here's this person when they first presented, they had significant disease, when first presented with COVID-19, after about a month, had even higher antibody titer. And then later, actually, I'm looking at the wrong, sorry, yeah, here, pre-COVID, I'm sorry, pre-COVID, low titer of antibody to RBD, then high immediately after COVID, and then that's beginning to diminish, but still significant. Person that had the mild disease, here it is, goes up a little bit. I think I actually show that, yeah, here a little bit easier to see in this graph. So that's for SARS-CoV-2, RBD, substantial increase in antibody response after having disease, and then it decreases over time. This person had milder disease, increases just a little bit, and this is the same sample run on a second plate, and just to look at plate-to-plate variability, it's minimal. This is the person's spouse. There's an insignificant change in the antibody titer, and then hospitalized patients, you can see have very high titer. Okay, so I want to take just a couple of minutes and tell you briefly about another technology. This tool, this arrayed imaging reflectometry tool, is a great way to get a lot of information in an array format. But because it's a free space optics format, we've got light source, chip, and then a detector. I'm never going to put this in a cell phone. If I want something that's going to be in a really small form factor, I have to go to an integrated photonics approach. And in the interest of time, I'm going to apologize and skip through this fairly quickly, but I want to highlight this as a way to make a very small, but yet very sensitive and efficient sensing technology that now is inexpensive, disposable, and potentially quite useful. So this work comes out of a consortium research effort that I've been leading through AIM Photonics, which is a public-private partnership to develop a manufacturing ecosystem for photonic sensors. This is something that the Institute was founded in 2015. We've been working with them through actually roughly 2015 and are just now starting to publish some of that work. But the idea is that if you're familiar with fiber optics, integrated photonics is what you get if you shrink those down to the size of a couple hundred nanometers on the surface of a silicon chip. And we make all of our structures in silicon nitride. It's a very good material for making silicon photonic sensors. Again, I'm happy to talk with you individually about that at another time. And so basically the way waveguides work is it's sort of like running water through a paper towel tube. Eventually the paper towel tube will get soaked and the water will sample the environment. If your hand is around the paper towel tube, you'll feel the water, but actually the water running through the tube will also feel the warmth of your hand. And the same thing is true with a photonic waveguide. Some of the light leaks out, it samples the environment and goes back in. And if you have a spectrometer or a detector at one end, you can actually detect that. That's how you make a sensor, particularly if you incorporate a modulator of some kind that can give you a spectral feature to observe. So we worked on a bunch of these. I'm going to focus on ring resonators, which are the same phenomenon with light that you may have encountered if you've stood in a dome like St. Paul's Cathedral Dome and whispered. Whispering gallery effect transports sound around the perimeter. Light will do the same thing in that if we have light going on a photonic waveguide, for most wavelengths of light that will pass all the way down the waveguide unaltered, and you can detect it. But for some wavelengths, namely those that can couple into a ring and circulate, ending up in phase back at the starting position, those will just circulate for a long period of time, and you'll see a dip in power at that particular wavelength at your output detector or spectrometer. The equation that describes this phenomenon of resonance is here. And so basically that resonant wavelength is the function of the geometry of the ring times the effective refractive index. And that's how we make this a sensor, in that something sticking to that ring changes the effective refractive index, and we get a shift in the resonant feature that we can observe. So we have developed methods for programming these. We fabricate them at a 300 millimeter wafer fabrication process at the AIM Photonics Foundry. Again, I won't go through all of this, but we're making lots of structures, including prototype on-chip spectrometers, Raman sensing systems, as well as the ring resonators. And again, at wafer scale. So here, and I'm going to have to skip over this, for the COVID sensors, what we do is we take these chips that are 1 millimeter by 4 millimeters, so roughly the same size as a grain of rice, and we take one ring, we deposit an antigen on that, covalently attached. We take the other ring, and we functionalize that with our anti-fluorescein molecule, and we just compare the shifts in these two rings. So that's easy to do using a piezoelectric spotting system. Thinking about scale, the spotter that we have, you could, if you had a way to effectively line them up on the platen, you could do 74,000 of these chips at a time. And then the assay workflow, you take blood, separate the serum, although it's actually not clear that we need to do this serum separation, and then apply this to the consumable. I apologize, I can't actually show you the consumable. This involves a proprietary microfluidic system that we're developing. But then we have this SARS-CoV-2 antigen that is barrel-shaped, our control. And as we look at this over time, we expect to see a shift in the SARS-CoV-2 antigen relative to the control. We can come back in with an anti-IgG if we want an IgG-specific response. And so here's what that data looks like. Here's from a convalescent COVID-19 patient. This person had a high antibody titer. These particular samples were diluted 1 to 5, so this is still a huge amount of antibody that's present. But what you can see is that here's the direct response for the two rings. This is the SARS-RBD, SARS-CoV-2 RBD response. You get that red shift, very easy to see. The response of the control is modest, but it's there for the anti-fluorescein. And then at the end here, this is starting to dry out in the fluidic system. If we come back and subtract that, here's what the response looks like. Okay, so very clear, strong response, where really we could declare this to be a positive at a minute or less. So it's a very fast assay. The IgG specific response, applying that then to this chip, we also get a response for that at a minute or so. And again, that can be quantitated once calibrated to a standard. For a negative sample, there's really no shift, either on the COVID-19 RBD protein or the reference. Subtract those, you get pretty much a 0, although again, you're running out of solution here. And then the IgG also gives a negative. So this is just a couple of examples showing that the assay works and works for different antigens. So this is another sample that had a slightly lower titer, and we can still pick this up, subtracted for RBD, or with another chip that's S1 plus S2, also quantitative, also quite fast. So this is an assay that I think is going to prove very useful as an alternative for that really quick, easy way to quantitatively detect specific antibodies to SARS-CoV-2 antigens. So to conclude, in the photonic side of things, and I apologize for how quickly I went through that, but this disposable photonics platform gives a rapid method for detecting antibodies to SARS-CoV-2. Actually, I can say this more definitively now. We've got enough data that this really gives us a quantitative amount that we can reference to antibody concentration. We've also developed a reader system. You know, one of the problems with photonics is the alignment problem. We've developed a system that lets us actually align in 10 seconds or less manually. We're automating this, so we think the alignment is going to happen in about a second. And so this is something that we can scale up literally to millions of chips quite easily. We're continuing to work with the AIR system, particularly in the Ziva format, to look at other samples. We have an IRB approval to go up to 2,000 samples. So we're extending this and beginning to look at how we might apply this to understand the disease and potentially contribute to vaccine development. Although hopefully, if some of the news that's coming out, maybe that'll be a solved problem, but I think we can still contribute in useful ways. So finally, I just want to thank the people that did the work. When we were on lockdown, Dan Steiner and John Cognetti were two students who stayed in lab and did all of the experiments. They would basically generate data, then hand off to people at home, particularly Alana Close and Ethan Luta, as well as Michael Bryant and Joe Buchkowski to analyze the data, and then the cycle would repeat. Had terrific collaborators and appreciate funding. And obviously earlier work, actually there's Alana, I guess I don't have a picture of Ethan, but the early flu work that I showed you, Hanyan Zhang, for those of you who haven't met him, that was his work, and it really is what made all of the subsequent work possible. So thank all of them, and I'll thank you once again for giving the opportunity to talk today, and happy to take any questions.
Well, thank you so much, Professor Miller. Very interesting talk. So floor is open for questions. I'm not sure, I can look at the chat. So if you chat a question, I know I'll be able to see it. I don't know if we'll be able, can a person unmute themselves and say a question? I'm not sure if we can do that.
I don't think anybody's allowed to.
Okay. So while other people are thinking of their questions, I have one for Professor Miller. So really interesting work, and just amazing what you can do with the multiplexing and seeing all the detail of the antibody repertoire that develops. So do you see this kind of test being used on, like people getting, COVID vaccine to tell how well a vaccine worked on individual people, or do you see this more as a research tool for evaluating vaccines on smaller numbers of people?
So I think there's really a couple of ways to look at that. I think assays like this, whether it's our assay specifically, are going to be really useful for looking at people post-vaccination to see how well the vaccine took. I mean, obviously, I'd like it if people are if ours is being used. But I think generically, these kinds of antibody tests are going to be super useful. And we actually saw that very early work that we did Charlie Mace, who's now a faculty member at Tufts, with the hemagglutinin array, looked at a vaccine trial, and we were able to very accurately assess whether people had an effective response to the vaccine or not. Obviously, we're not getting at neutralization with this. We're just looking at binding. But as it turns out, binding is actually a pretty good surrogate for neutralization. There aren't too many tight binding antibodies that aren't neutralizing. And there's actually good work now with that in SARS-CoV-2 as well. On the research side, to me, what's really interesting on the research side, particularly with the array work, is starting to look at these correlations. How does immunity to other upper respiratory viruses impact somebody's response to SARS-CoV-2 or flu or what have you, right? How does that all relate? And how does it change over time? So the correlation bit is something that I'd expected. I hadn't thought about how interesting it's going to be to follow people over time who've had COVID-19 and just watch how their immune system changes. But I think that's turning out to be a really interesting research tool.
Thank you. More questions. I'm watching the chat, see if anyone types one in. I don't see any yet. Okay, so I guess Professor Miller, you're off the hook for more questions.
Okay.
But I know also it's after the hour, and I think a lot of people moving on to their next Zoom meeting of the day. But thanks so much for sharing your work. It's really exciting, and just wish you all the best of success. And I hope that this makes an impact getting people back to work when we can get immunized.
Well, thank you, and thank you again for hosting me. And it's been delightful talking to a few of you through the day. And again, I'll look forward to meeting again in the real world at some point.
Yeah, absolutely. I look forward to having you visit, like for real.
Right. All right. Thanks.
Well, thank you so much.
Bye.